Please welcome Dr. Gordan Lauc to the A4M stage today. Dr. Lauc is the Professor of Biochemistry and Molecular Biology at the University of Zagreb and Director of the National Center of Scientific Excellence in Personalized Healthcare. He initiated the launch of the Human Glycome Project and is one of its co-directors. He is a president-elect of the International Glycoscience Organization and member of the steering committee of the European Glycoscience Forum. Dr. Lauc is also the world's leading glycan researcher. His research team is pioneering high-throughput glycomic analysis and the application of glycan biomarkers in the field of precision medicine. He's an accomplished author and founder of Genos, a biotech company that is currently the global leader in high-throughput glycomics. Welcome to the stage, Dr. Lauc.
So, good morning. It's a great pleasure to be here, to have a privilege to introduce all of you to the world of glycans, a field which is probably not very familiar to many of you. And is sound okay? So the question which I will ask and try to answer today is, can we make a decision to stop or even revert the glycan clock of aging and slow down inflammaging? But before I start, I have to disclose that in addition to being professor at University of Zagreb, King's and Edinburgh, I'm also founder and owner of the Glycans Glycoscience Research Laboratory, the leading glycomics lab in the world, and also the chief scientific officer and co-founder of the Glycan Age, which are commercial companies active in this space and also have a number of patents. So I'm commercially involved in the topic that I will be talking about. So, glycans. They're the ultimate layer of molecular complexity. The majority of proteins are composed not only of polypeptide part of the amino acids, but they also have this glycan coating. Actually, majority of extracellular proteins have more glycans on their surface than amino acids. And the analogy I like to make are these two birds. So the polypeptide part is the flesh and bones of a bird. The bird cannot function without its polypeptide part. And many scientists focus their research on the polypeptide part. And you can do a lot of good science on polypeptides. You can study the physiology of that bird. You can study even the walk of that bird. But you will never understand flight, because bird without its feathers cannot fly. Protein without its glycans cannot function normally, in particular on the cell membrane. The lipid bilayer we learned in school is this tiny little white lane, while the functional cell membrane are the glycans, the glycocalyx on the surface on the cell membrane. And the majority of proteins are glycosylated. And to understand biology, to see the complete picture, we have to look into glycans. But the word of caution here, glycation, glycosylated hemoglobin, HbA1c, advanced glycation end products, this is something completely different. Glycation is the random damage of glucose reacting with amino groups on a protein. It's like taking a shotgun and firing on a protein. Glycosylation is a sophisticated modification which makes protein better. Actually, I believe that the invention of glycosylation was the third revolution in evolution, which enabled us to become multicellular. The first revolution was the invention of nucleic acids, the second were proteins, and the third were glycans. And the key difference between the glycans and other components of life is that there is another layer of complexity. So our proteins are defined by our genes. So there is a gene sequence determining the sequence of amino acids and the structure of a protein. But the glycans, which are also part of a structure of a protein, are encoded in a complex network of dozens of genes which work together to make a decision which kind of glycan to produce and decide about the structure and function of a protein. An alternative glycosylation, so the adding different glycan structures to the same glycosylation site, is functionally analogous to the coding mutations. And we know that mutation can kill us. An alternative glycosylation can also do either a good or a bad to a protein. And in a study we published a couple of years ago in the Nature Chemical Biology, we very clearly demonstrated by reshuffling the genetic variants in creation of a collaborative cross-cohort of mice. So what Grant Morahan did, he took 8 founder strains of mice, crossed them for 3 generations, and then inbred them into strains. And some of these strains, as you can see here, became very different. So the mice became very different in their structure, in their function, without any mutations, just by reshuffling the allelic variants. And this is also what we see in humans. It is actually glycans which are responsible for a large part of inter-individual variability. So the reason that we are all not the same are by large glycans. Even blood groups, A, B, and O blood groups, are chemically glycans. This is a study we did, I think, nearly a decade ago, just showing how people are different. So these are the glycans attached to immunoglobulin, different glycan, different color, 6 random individuals. Just by looking at it, you can say people are different. And now you can ask yourselves, so why did you hear about it before? So why are not more people working in the field of glycobiology? And my frankest answer is, I don't know. Because there are very strong policy documents, both in Europe and in USA, for nearly 2 decades, which claim glycans are directly involved in the pathophysiology of every major disease. And the additional knowledge of glycoscience will be needed to realize the goals of personalized medicine. Maybe the reason is that glycans are very complicated structurally, and that their analysis is challenging. So to a backbone of polypeptide, which forms a protein, different glycan structures can be added to different glycosylation sites. So the level of complexity is enormous. And to analyze it, we need to perform many chemical steps, and there are actually not so many laboratories which can do it, and especially not in high throughput. And at the moment, the glycoproteome can only partly be characterized. We cannot sequence entire glycoproteome. We cannot determine the entire structure. We can just look at tiny bits and pieces and analyze either the released glycans or the glycopeptides and get some insight about the glycobiology of life. But the progress is rapid. 15 years ago, we were studying a couple of patients. Now we can do studies with more than 10,000 people in a single study, which enabled glycomics to merge with the other omics. And actually, even this year, the Nobel Prize in Chemistry was awarded for the development of methods of glycan analysis. Caroline Bertozzi from Stanford got it for her click chemistry and analysis of function of glycans on the membranes. My lab, GANOS, is the global leader in high-throughput glycomics. We analyze more glycans than anybody else. Actually, the recent study in Chemical Reviews revealed that out of 191,000 published glycoms, we generated approximately 160,000. So nearly 80% of the global glycomic output comes from GANOS. And we did it in collaboration with some of the best researchers all around the world. So we were not analyzing any people. We were analyzing people from the best clinical epidemiological cohorts to gather as much data as possible, which then enabled us to move the field of glycomics forward. And actually, now we routinely publish in some of the leading journals, including the Gastroenterology, Circulation Research, Diabetes Care, and so on. So everything what I will be telling you now is hard science. And despite the fact that I do have commercial interests, I will be very careful to disclose them each time and show you only the hard, verifiable things which are published in good papers. So one thing we are very interested in, all of us, is inflammation. Inflammation is good when it is under control, but as we are getting older, there is more and more low-grade chronic inflammation, which is actually underlying the majority of complex diseases of the modern world. And glycans are here very important, especially glycans attached to immunoglobulins, because these structures can make immunoglobulin molecule either pro-inflammatory or anti-inflammatory. So something which people often forget, IgG is one of the most important anti-inflammatory molecules we have. If we give IVIG therapy to a patient, we will suppress inflammation, mostly by these inflammation-suppressing glycans. And something what happens with time is that people are losing anti-inflammatory glycans and are getting more and more pro-inflammatory glycans as we are getting old, and this is driving inflammation, but not only at the level of IgG, but at the level of many other cells and processes which are involved in inflammation. And we know that inflammation is one of the main evolutionary conserved pillars of aging. And it's also shared by the age-related diseases. So we have a stem cell regeneration, metabolism, proteostasis, molecular damage, stress, epigenetics, all working together to either control or to promote inflammation. And as a recent cover story in the New Scientist said, we are only as young as our immune system. And if we can reset our immune age, we can definitely slow down aging, at least the inflammaging component. But we do not know what is going on in each of us. Especially, we do not know what is going on in a patient when you see them for the first time. And something what we learned from our studies, and we analyzed hundreds of thousands of people, is that as we are getting older, glycans which promote inflammation increase, and those which suppress inflammation decrease. So what we see here is each row is a different glycan structure. Each column is a one large cohort of people of at least 1,000 individuals. Blue are men, red are women, and we see that everything changes in the same way in all cohorts, and pro-inflammatory structures go up, and anti-inflammatory structures go down. And when we published this first time in 2013, editors of the journal wrote an editorial asking, are glycans the holy grail of the biomarkers of aging? And actually, I think they were right. I really believe that the glycans are the holy grail of the biomarkers of aging because they are not only biomarkers, they are functional effectors. Because something can be a biomarker, but if you change it, nothing happens. But if something is a functional effector, when you change it, there has to be a change in an outcome. And this test is today available. Glycans can be measured. Structures can be quantified. We know that this integrates environment, epigenetics, genetics, stress, sleep, smoking, everything integrates into the changes of these glycan structures, which regulate IgG functionally, and this is something which can be measured. And of course, I have to repeat here, I'm in a conflict of interest, of course. This is a company which I founded, and I have I'm also inventor of the patent for this test. But fortunately, other people do the same things and show the same things as we did. So a large study done in Weill Cornell, Qatar, they measured nearly everything what can be measured in their cohort. They measured over 6,300 different molecular traits, including the both O-link and somologic panels for the proteomics, metabolomics, lipidomics, epigenetics, genetics, everything what can be measured in the people they measured. And then we looked when they associated with age, and then they just ranked the best associations out of these 6,000 associations. 9 out of 20 associations were glycans attached to immunoglobulins. And the other 11 were mostly sex hormones, which we know they change a lot with age. So I believe that today the glycan test of biological age is already established as the best personal navigator of healthy aging, mostly in Europe, because we are Euro-based, but it was featured in all major newspapers. We were on a headline of the Wall Street Journal for, I think, 3 days as one of the most read stories. So this is definitely something which is becoming appreciated. I'm just a scientist. I'm not heavily involved in these companies. Actually, I'm nearly not involved at all in any commercial aspect. So what I'm trying to do, I'm trying to understand what is going on, how is this being changed, and what are the functional consequences of changes in glycans. And the first thing to start, of course, is genetics, because it's so easy. We sequence tens of millions of people, even hundreds probably. And in most of our patients and participants, we had genetic information. So we did a series of so-called genome-wide association studies where we looked at the links between the glycans and the genes, trying to understand which genes regulate IgG glycosylation. And through a series of papers in the last decade, we mapped a network of over 40 genes which are involved in IgG glycosylation. And only a few of these genes, these orange on the end, they're actually genes which synthesize glycans. They're the enzymes which make the glycan structures. All other genes are some kind of regulatory molecules which make a decision how to glycosylate a protein. And these genes are known risk factors for 94 different phenotypes. So we did so-called pleiotropy analysis. So we looked which genes both regulate IgG glycosylation and are known risk factors, and identified 94 different phenotypes, including some which are kind of expected: type 1 diabetes, lupus, Crohn's disease. We know inflammation is very important there. But also things like Parkinson or dementia or HDL or cholesterol concentration are somehow pleiotropic with genes which regulate IgG glycosylation. And this is protein specific, because in a study published in Nature Communication earlier this year, we showed that despite the fact that the glycans are nearly identical, genes which regulate glycosylation on IgG and transferrin are nearly completely different. So the enzymes are the same, the structures are the same, but they're added in the context of different proteins and in the context, of course, of different cell, either the hepatocyte or the B cell, actually the plasma cell, and the regulatory network is completely different. So we have a protein-specific, complicated regulatory networks which decide about protein glycosylation. And on top of that, we have epigenetic regulation of all of these genes, because genes can have different activity because of sequence variation, but genes can also be silenced by epigenetics, especially through time. And there's also direct environmental factors affecting the activity of genes. So we have a genetic component, we have epigenetic component, and we have environmental component integrating into a chemical structure of a glycan attached to a protein, in this case immunoglobulin G, deciding whether this protein will promote inflammation or suppress inflammation. So when we looked in the world how different people are, we asked our friends from all around the world, 27 different cohorts, to send us plasma samples. We analyzed IgG glycans, and we compare them, and we see people are different. People in different regions of the world are different. And when we tried to see what explains this difference, the strongest predictor was the expected lifespan and the Human Development Index. So people living in rich countries had a longer expected lifespan, and their glycans were changing from anti-inflammatory to pro-inflammatory much slower than people living in the countries with a shorter expected lifespan. And approximately 40% of that is genetic. So we did a large study with over 10,000 twins to see the heritability of the glycan age clock of aging, and the heritability is around 40%. And the remaining 60% is a lifestyle. So people living unhealthy lifestyle will have accelerated glycan aging, while people living a healthier life will have slower glycan aging. So this acceleration of glycan age correlated with the lifespan. Interestingly, the acceleration in the glycan aging did not correlate with acceleration in epigenetic aging. This was study done on the University of Edinburgh. It was not our study. So they compared all the different clocks of biological aging, and they found that although glycans correlate with some kind of other metabolic clocks of aging, the acceleration in glycan aging and epigenetic aging did not correlate much. So obviously these are two different aspects of aging. Epigenetics is something within the cell, while glycans are integrating aging at the level of an organism. Another very important aspect in the acceleration of glycan aging are different diseases. We studied a couple of dozen patient control cohorts, and in most of them, we see the effect of changes in glycans to be very similar to the effects of aging. So the last 4 bars here are just population cohorts. This is effects of aging, comparing old to the young people. All other bars are different diseases, including lupus, ulcerative colitis, Crohn's disease, arthritis, GVHD, COPD, type 2 diabetes, colorectal cancer, HIV, and so on. And glycans, which are part of the glycan age clock of aging, and this is the pro-inflammatory, these are the anti-inflammatory glycans, and this is also anti-inflammatory glycan. We see that these changes are practically going in the same direction as aging in the majority of diseases. Not all of them, but in the majority of them. And what we believe that we see here, actually, we see the functional role of IgG glycans, which modulate low-grade chronic inflammation, and in this way participate in the development of many different diseases. So these are not disease-specific changes. These are inflammation-specific changes, which we see in multiple different diseases. And the third very important aspect of changes in glycans are the sex hormones. We published this first time 5 years ago, where we showed that it is estrogen which very strongly affects IgG glycosylation in both men and women. So we got access to a very nice placebo-controlled randomized trial of both men and women on the hormonal suppression and then either supplementation with testosterone and estradiol or the placebo. And we see that the placebo group aged on average 9 years within 6 months, while the estrogen supplementation did not. In men, it was again estrogen. Testosterone was given, but if the aromatase was blocked, then there was no effect on the improvement of glycosylation. So if you're trying to keep testosterone very high, don't use aromatase. It's blocking the beneficial effect of estrogen on IgG glycosylation. And in a number of studies, we have also shown that these changes in glycans happen up to a decade before people are diagnosed with diabetes, cardiovascular diseases, inflammatory diseases. So glycans change first, and a diagnosis comes later. And this is not surprising, because diagnosis is actually an endpoint. It's very often beyond the point of no return, because we keep on going, and only when something breaks down, we ask for help, we got the diagnosis, and usually then we get some kind of pills which keep us alive, but keep us ill. The molecular process leading to the disease is very long, at least a decade-long process, where some molecular changes are visible, which eventually end up in a failure of some of the organ systems. And glycans definitely play an important role here. So the general idea is that we don't wait for people to become ill before we do something. We try to find them here. We've tried to find the first indication that some of the molecular effectors which lead to disease are already disturbed, and then we can make an intervention, and we can either postpone or completely prevent development of a disease. And I believe glycans can really help a lot here. One example, cardiovascular disease, the main killer. Most of us will die because of cardiovascular problems. Initial study we did was published in Circulation Research 4 years ago. We studied approximately 3,500 people in England and Scotland, and we realized that IgG glycan composition associates with the cardiovascular disease risk score. So the glycans to date associate with a predicted risk of diseases in the future. And we used that data to persuade people sitting on the committee of the EPIC cohort, and the EPIC cohort was collected 30 years ago, samples are stored in the fridge, and there is a clinical follow-up. And we got access to the German cohort of 27,000 people, and out of these 27,000 people, there are around 400 of them who developed either heart attack or stroke, and we looked into glycans 10 years before, and we saw that the predictive power, the ROC curve, just for these few glycan structures which we measure, were as good as the entire AHA score is in predicting the cardiovascular disease events. And we went to replicate it, and this was just reported as a poster on the last AHA conference. It will be published soon, I hope. A large study on the 2 cohorts together with Professor Samiya Mora from Harvard, and we have shown very clearly, again, that IgG glycans associate with the future cardiovascular disease events independently of other clinical risk factors. So there is something in glycans which make people more prone to future cardiovascular events. And we believe there is a molecular mechanism because there is an independent line of research showing that these anti-inflammatory glycans, the glycans with the salicylic acid which are suppressing inflammation, are also binding to the Fc gamma receptor 2b in endothelial cells, and through a signaling mechanism promote release of nitric oxide and promoting vasodilatation, while the old glycans cannot do that. So there is a way how young anti-inflammatory immunoglobulin glycans can also affect the endothelial biology and protect for cardiovascular events. And in an animal model, we can actually rescue that by improving IgG glycosylation. So this was a study done by Phil Scholl at the UT Southwestern. So he was feeding mice with a high-fat diet. Mice became obese, and they developed hypertension. And they lost these young immunoglobulins. They didn't have the young immunoglobulins. They have these old immunoglobulin glycans. And then he was feeding mice with a dietary supplement called MANAC, which is N-acetylmonosamine. Molecule very similar to N-acetylglucosamine, just it is not commercially available because there is no interest at the moment. And these mice on the MANAC did not lose young IgG glycans. They did save these young immunoglobulins. Of course, they became obese, but they didn't develop hypertension. So using a dietary supplement which improves IgG glycosylation, obese mice can be protected from hypertension. For humans, we do not know. There were 2 small trials on NIH showing that N-acetylmonosamine is safe, but there are no studies in obese people to see whether they will be protected from hypertension. What we did, we looked at the large cohorts, and we did see that the people with hypertension have older glycans than people without it, but this is still not proof of causation. Also, there is a correlation between the accelerated glycan aging and the future type 2 diabetes. These pro-inflammatory IgG glycans are also predictive of both prevalent and incident diabetic complications. people with the older IgG glycan were at higher risk to develop more severe COVID than the people with a younger IgG glycan, which is kind of logical because it's the biological age which is the risk factor and not the chronological age. And also, people with a severe COVID were aging much faster than people with a mild COVID. So there are many people who with a moderately severe to severe COVID aged 5, 10, 15 years within a couple of weeks when they had COVID. So to come to the key question, can we revert it? So what can we do to revert the glycan age clock of aging and stop inflammaging? So one of the best advantages of the glycan age clock compared to other biological clocks is that it's the response to interventions which are known to be beneficial for health. This is one of the key advantages of the current epigenetic clocks. Maybe some of the future epigenetic clocks would be better in this respect, but generally it's very difficult to change them. While for glycan age, there is a lot of evidence showing that you can actually change it with beneficial interventions. For example, many anti-inflammatory drugs will move the IgG glycan to the anti-inflammatory direction. This was a large study on biologicals, infliximab, adalimumab, and a couple of others, which would make IgG glycan less pro-inflammatory. Of course, you will not use this type of drugs in any kind of a preventive setting, but also simple things like a weight loss or a weight gain would have a very strong effect on the IgG glycan composition. We were tracking 2,000 twins for 15 to 20 years with sample collected in 3 time points. So in these nearly 2 decades, some of the people were gaining weight, some of the people were losing weight. So we just looked at the BMI. So BMI is not the ideal measure when you are comparing different people, but when you're looking at the same person through time, BMI is generally a good indicator of obesity because very few people, especially in older age, will gain mass in the muscles. Mostly we gain just fat. And when we looked at these 3 sets of people, there were some who were the same weight from their 30s to their late 60s. Some of them were gaining weight, and some of them were losing weight. And people gaining weight were aging faster in all time frames we looked at. So in their 30s, in their 40s, in their 50s, they would be aging faster if they gain weight. If they lose weight, they will be aging slower, sometimes going even in the reverse. If there was a drastic intervention like a bariatric surgery, in many people, this was followed by the radical decrease in glycan aging. We had one patient who improved for 37 glycan years in 6 months after bariatric surgery. Not everybody moved so much. Some even moved in the other direction, but the majority of people, there was a very strong anti-aging effect of weight loss. And caloric restriction also works in the majority of people. This was studied on 1,000 people on a very strict low-calorie diet done in, I think, 8 centers all around Europe. And in 2 months, a low-calorie diet, they all improved. But the low-calorie diet is not a solution. It's a torture. You don't want to be hungry all the time. So the study then looked into 5 different maintenance diets. So different combination of proteins, glycemic index, and so on. And what we learned here is actually is something what we knew from the beginning, and this is that we are all different. And there is no one solution which will work for all of us. And I think this is one of the reasons why all the recommendations change so drastically, especially in the field of diet, because some type of diets would be good for some people, the other type of diets would be good for other people. And I think that the frankest answer is we don't know what will work for each of you until you try and see whether it works. And Glycans give an opportunity to actually try something and a few months later see whether this specific type of diet was working for somebody or not. And The same goes for exercise. We have been cheated for decades that you can sweat out the bad diet, that obesity is not a problem of a junk food, it's a problem of you being lazy. This is false. We cannot sweat out the bad diet. Actually, when we started using glycans to look at different interventions, we first focused on gyms and different types of physical activity. And we actually learned that majority of people overtrain. Especially if we take middle-aged people and tell them, you have to be active, you have to train, you have to improve. They will overtrain, they will go into pro-inflammatory cascade because of too much physical activity. And the physical activity has to be optimized to a given patient and balanced with a sufficient relaxation periods. Another extremely important element is microbiome. Changes in microbiome are changing the glycome, and at the same time, glycome is affecting the microbiome. We have a couple of studies on microbiome transplant. We have a couple of randomized trial now, very cool randomized trial in personalized dietary intervention also going on at the moment, hoping to understand the complex relation between diet, microbiome, glycome, and health. So this was all science. So what about individual people? So how does it look at individual level? One famous example is Tim Spector. You probably know about him. He's one of the most cited scientists in the world, author of many fantastic textbooks. We collaborate for over a decade. His Glycan age was good for a number of years, and then stress, too much work, he got worse, and he decided to do an intervention. We actually recorded Instagram Live, and he just published a book saying what the book is a spoon-fed, what he did. But he managed to lose 11 Glycan years in less than a year. So things are doable. One of the things he did was a metformin, but I don't like to claim that anything works until we see a really good placebo-controlled randomized trial to identify which of the things are actually functioning. There are some clinics, and there are now over 600 clinics selling the Glycan age globally, who are very successful in reducing the Glycan age of the patients. Joe Raffaele is one of the speakers on this conference. Most of his clients become 15, 20 years younger after treatment. Menopause is extremely promising field for the Glycan research, because the problem with menopause is that it's so rapid in women when you talk about the Glycans. So while women are mostly look younger than men in their reproductive age, there is a huge acceleration of Glycan aging, which happens before any other symptoms of menopause. And so this is the premenopausal woman, perimenopausal woman. The rate of Glycan aging at least decreases 4 times or even more in this short period of transition. And this happens usually before any other symptoms of perimenopause. So what we see with our clients is that somebody would be fantastic in their Glycan age, and then this particular lady aged 30 years within a year. And of course, she freaked out because there were no other symptoms. She said, I'm not in menopause. But then with the optimal hormone therapy, these things can be reversed and can go back in the favorable direction. And actually, we believe that in a couple of years, IgG glycan composition, which can be measured by the Glycan age, in the field of perimenopause will be the same thing as HbA1c in the field of glucose, because HbA1c integrates the glucose concentration in the last couple of months. IgG glycan integrates the estrogen concentration in the last couple of months. And this is now also important in men and in women. And I'm out of time, so I will skip the last few slides. I will just say here, analogy I like to use is, we treat our body as something which has to be fixed only after it explodes. Imagine that we're changing our tires only after they explode on a highway. Not very healthy approach. Glycan age can help to motivate people to see whether what they're doing is good or not. And I have to finish with acknowledging all the funding agencies. We got many research grants to do it. And we have no time for questions, so if you want to ask any questions, I will be close to booth 9033, where the Glycan has a small booth, and you can send me an email for any questions. We are very collaborative and like to collaborate with people with good clinical samples. Thank you.